Hello, my dear students. Welcome back to another learning lessons in my Physio-Pulse channels. Today we will discuss about the gluconeogenesis process. Actually, the definition of gluconeogenesis and the site of gluconeogenesis and the total steps of gluconeogenesis process. First of all, gluconeogenesis is the formation of glucose from non-carbohydrate sources. These sources includes the lactic acid, lactate, pyruvate, glycerol, propionic acids, amino acids, like the glucogenic amino acids. Where it occurs, actually gluconeogenesis location, subcellular location is cytosol or mitochondria, and the cellular or organic location is liver 90% and kidney is 10%. So the process glucose is converted to pyruvate or lactate, that is glycolysis. On the other way, if we see pyruvate or lactate, which is reconverted to form the glucose, is called the gluconeogenesis. Gluco means sugar, neo means newly, and genesis means synthesis or create. That is the newly synthesis glucose is called the gluconeogenesis. So now to see how the process is occurred. As we know, the glycolysis pathway, that is the glucose is converted to form the glucose 6-phosphate with the help of hexokinase enzyme. Then glucose 6-phosphate will convert to form the fructose 6-phosphate with the help of phosphofructokinase, PFK. And it is a rate limiting enzyme. This PFK, it will convert, it will form the fructose 1,6-phosphate, and then subsequently with the aldolase enzyme, it will splits to form, that is glycerol 3-phosphate and dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate and glycerol 3-phosphate, what happened? This glycerol 3-phosphate, it will convert to form the 1,3-bisphosphoglyceric acids, then ultimately 3-phosphoglyceride, then 2-phosphoglyceride, and phosphoenol pyruvate will form. And now pyruvate kinase, with the help of this enzyme, phosphoenol pyruvate will convert to form the ADP to ATP. This is the whole glycolysis summary of the glycolytic pathway. But the gluconeogenesis reactions, the synthesis of glycolysis from pyruvate utilizes many same enzymes like the glycolysis. And three glycolytic reactions are essential, which are irreversible. What are these enzymes? Already we have discussed hexokinase, PFK, this is the hexokinase, and PFK phosphofructokinase, and then pyruvate kinase, these are third enzymes. These three stages bypassed by four enzymes specific to the gluconeogenesis. They are called the key enzyme for the gluconeogenesis. The pyruvate carboxylase is first number 1 enzyme, and then phosphoenol pyruvate carboxykinase, then fructose 1,6-bisphosphate, then glucose 6-phosphate. So if you see the bypass mechanism, there is the first bypass reaction, that is the conversion of pyruvate to phosphoenol pyruvate. How pyruvic acid is converted from the phosphoenol pyruvate, and that we will discuss in the first bypass reaction. It requires the participation of both mitochondrial as well as the cytosolic enzymes, and pyruvate is transported from the cytosol into the mitochondria via the mitochondrial pyruvate transporters, or pyruvate may be generated within the mitochondria via the deamination of alanine. Now pyruvate is converted to oxaloacetate by the biotin requiring enzymes, that is pyruvate carboxylase, which is as follows. Pyruvic acid plus HCO3 minus plus ATP, it will convert to form the oxaloacetate ADP plus PI. And this pyruvate carboxylase is the regulatory enzyme. Now oxaloacetate is reduced to form the malate, and with the in the mitochondrial malate dehydrogenase enzyme, and at the expense of the mitochondrial NADH. What happens? The reaction is oxaloacetate plus NADH plus H plus. It will form the L-malic acid or malate and NAD plus. Malate exists in the mitochondria via the malate or alpha-ketoglutarate carrier. In the cytosol, malate is reoxidized to form the oxaloacetate via the cytosolic malate dehydrogenase with the production of the cytosolic NADH. Then malate plus NADH, it will form the oxaloacetate plus NADH plus H plus. Now then oxaloacetate is converted to form the phosphoenol pyruvate by the phosphoenol pyruvate carboxylase enzyme. So this is the whole overall reaction: pyruvic acid plus ATP plus GTP plus HCO3 minus, it will form the phosphoenol pyruvate ADP GDP plus PI plus H plus CO2. So the synthesis of one molecule of phosphoenol pyruvate, which requires investment of the one ATP molecule and one GTP molecule. So one important thing is that is when either pyruvate or ATP or ADP ratio is high, the reaction is pushed towards the right. That means the direction of biosynthesis is happening. So the summary of the first bypass reaction is that in mitochondria, where pyruvic acid is converted to form the oxaloate state with the help of pyruvate carboxylase enzyme, then this oxaloate state, which is again converted to form the malic acid or malate with the malate dehydrogenase and NADH as a cofactor. Now this malate comes to the cytosol, and where it with the help of malate dehydrogenase enzyme and NAD plus, it will convert to form the oxaloate state, and now it will convert to form the phosphoenol pyruvate with the help of phosphoenol pyruvate carboxygenase enzyme. Here one molecule of carbon dioxide is reduced and one GTP to convert to form the GDP. So second bypass reaction, what happened? The conversion of fructose 1,6-bisphosphate, which will convert to form the fructose 6-phosphate. The third glycolytic reactions, which is irreversible and hence the gluconeogenesis, fructose 6-phosphate must be generated from fructose 1,6-bisphosphate by different enzyme, that is fructose 1,6-bisphosphatase. This reaction is irreversible also. Now fructose 1,6-bisphosphate plus H2O, that will reach to form the fructose 6-phosphates plus PI. Now third bypass reaction, glucose 6-phosphate, which will convert to form the glucose molecule. Because of the hexokinase reactions, which is irreversible, the final reaction gluconeogenesis is catalyzed by different enzymes, namely glucose 6-phosphatase. Now glucose 6-phosphate plus H2O it will forms the glucose plus PI. Now glucose 6-phosphatase is present in the liver but absent in the muscles. Thus the glucose produced by the gluconeogenesis in the liver is taken by the bloodstream to the brain and muscles. So summary of the gluconeogenesis pathway is that here the red color shows in the enzyme of the gluconeogenesis, and the blue color which shows the enzymes of the glycolysis. So in this way, here we see that phosphoenol pyruvate carboxylase from phosphoenol pyruvate to form the oxaloate states, and this oxaloate state also comes from pyruvate, that is pyruvate carboxylase enzyme. Now this phosphoenol pyruvate, it will leads to form the 2-phosphoglyceride, then 3-phosphoglyceride, 1,3-diphosphoglyceric acids, and ultimately it leads to form the glyceraldehyde 3-phosphate. Now if you see the glyceraldehyde 3-phosphate, when it combined with the dihydroxyacetone phosphate, then it will form the fructose 1,6-bisphosphate, and ultimately this fructose 1,6-bisphosphate, it will convert to form the fructose 6-phosphate and then glucose 6-phosphate, and ultimately it will form the glucose. So what we see from pyruvic acid, or that is the non-carbohydrate, it will convert to form the glucose in this way. This is the process of gluconeogenesis. So the overall reaction is 2-pyruvate plus 4-ATP plus 2-GTP plus 6H2O plus 2-NADH. It will form glucose plus 4-ADP plus 2-GDP plus PI and 2-NAD plus 6H. So for the conversion of 2 molecules of pyruvate into 1 molecule of glucose, 4 molecules of ATP and 2 molecules of GTP and 2 molecules of NADH plus H plus are utilized here. So this is overall reactions, and this is, I think, total steps or all the steps of the gluconeogenesis process which is occurred in cytosol as well as mitochondria, that we have discussed here. And I think students, if you have any query, any doubt, please give some comments in our comment box. And if you found this video helpful, please like the video, subscribe to the channel, and turn on the notification bell so you do not miss any biology lesson. Thank you for watching, and see you in our next class, that is the specifically with precursor of the gluconeogenesis process and the importance as well as the functions of the gluconeogenesis, that we will elaborately discuss in our next lectures. So thank you students.